Good morning, guys. The book I did my presentation on is called Epigenetics, How Environment Shapes Our Genes by Richard C. Francis. Hope you enjoyed the presentation. Here's the cover. We've talked about this concept in class before, although we didn't use the word epigenetics, about heredity versus environment. The book focuses on four main concepts: the basics of epigenetics, the stress axis, theories that can lead to epigenetics, and X inactivation. NIH gives a simple definition that epigenetics is changes in gene activity not based in actual gene alteration. This is done through gene regulation, which often shows up as a mutation. Like we talked about in class, your genes might code for a certain trait, but regulation of that gene might change the expression of it. The environment is a huge catalyst for changes in gene regulation. After all you inherit is your genes. The environment does the rest. An avenue for epigenetics to happen that the book focuses on a lot is methylation. So these red strings are going to represent our methyl groups, and this long helix is going to represent a gene or DNA molecule. The gene right now is not methylated at all. And now you can see that there's just a few methyl groups on the gene. This is called being undermethylated. And when a gene is undermethylated, it's overexpressed. However, when a gene looks like this and it's got a bunch of different methyl groups on it, it's called overmethylated, and the gene is underexpressed. This is because methylation acts as a blocker, as a repressant for gene expression. When there's all this methylation on a gene, it becomes very, very hard to translate and transcribe it into proteins, which is how genes are expressed. Being more or less tightly wound around histones, a type of genetic material that helps form the building blocks of a chromosome, can help gene expression as well. When we're born, we start with a mostly clean epigenome. Notice the mostly. Certain imprinted methylation patterns can carry on through a generation to affect the next one. This is just a picture of the chemical formula of what methylation would look like on a DNA molecule. And here is a diagram of what the histone wrapping looks like. As the center of four whole chapters, this book focuses a lot on the stress axis and how it's affected by epigenetics. The stress axis is a process by which the hypothalamus releases stress hormones called glucocorticoids, the most common of which is cortisol. Under stress, the hypothalamus releases corticotropin-releasing hormone, which is called CRH. This triggers the pituitary to release corticotropins, called CTs, which stimulate the adrenal gland to release glucocorticoids, which are stress hormones. The process shown in this picture, taken from the book, is the process of creating cortisol rather than just a general stress hormone. Cortisol is a steroid and can alter gene expression by bonding with its nuclear receptor. Womb environment has a large effect on the stress axis. When the mother has prolonged stress, the cortisol she produces is transferred to the fetus through the placenta in much higher doses than normal. Because of this, the baby's stress axis is rewired to be hyperactive and oversensitive. This is called HPA programming. Because of this, mothers with PTSD have children who are at more risk for developing PTSD. Back to the heritability of epigenetics and how it plays a role in the stress axis. In World War II, the Nazis took hold of the northern part of the Netherlands. This cut off their food supply and sent them into a famine. Clement Smith acquired OB records from the time and was the first to discover that prenatal nutrients are directly correlated to birth weight. This seems like common sense now, but it was revolutionary information at the time. This cohort of children born during the famine has been studied over the years as they've grown up, and they were found to be more likely to develop obesity and psychological conditions like depression and schizophrenia. Because these changes were caused by epigenetics, and epigenetics is in part heritable, their kids and grandkids were also discovered to have higher risks for these adverse effects of the famine, which is why it's known as the grandmother effect. Of course, after you're born matters too. As a form of care, rat mothers lick their young. Attentive mothers, good lickers, produce a rat that has a dampened stress axis. Poor mothers, bad lickers, produce a rat that's stressed for life. When pups are fostered by the opposite environment, they take on the response expected from the foster parent. Michael Meany and his colleagues at McGill University discovered that the rats with mothers who licked them more had a greater number of glucocorticoid receptors, which we'll call GRs, in their hippocampus, and were able to create a more sensitive feedback loop when it came to their stress response. So, if you don't remember what a negative feedback loop is, think of it as hovering around your body's homeostatic point. Your body will produce so much of a substance, and when the levels are too high, it stops producing and it goes back down. When the levels are too low, it goes back up again. And so it just looks like a wave around this homeostatic line. Turns out, when the GR gene is demethylated, a transcription factor called NGF was able to bond better to the gene, which increased GR transcription, hence epigenetic alteration of this gene. The book also goes into 3 different theories regarding genetics and how epigenetics plays into them. The first, preformationism. In the simplest terms, the zygote contained you. Our present selves are just a matter of growth, and everything about you has been there all along. The simplest way to explain the basics, which is the example the book uses, is that Eve's eggs contain the eggs of the next generation, and those eggs contain the eggs of the next generation, and those eggs, and so forth. More sophisticated theories for preformationism say that however latent and underdeveloped, you are still you. The book says, your development is the process of, quote, latent you becoming, quote, manifest you. One so-called virtue of the theory was that it didn't rely on what this book calls spooky supernatural stuff to explain how you came to be from your mother and how she came to be and so on. You, in a sense, have always existed as far back as our oldest ancestor. This doesn't take much to prove wrong. One famous experiment was done with sea urchins. 2 scientists named Roux and Driesch set out to discover how the complexity of a living organism resulted from the simplest zygote. Roux had a theory that after each subsequent division of a cell, chromosomes were parceled out, and thus arose the cell differentiation we see. They both used sea urchins, but Driesch was the only one to master the technicalities of the experiment. Driesch found that when he split the zygote at the 2-cell stage, the 4-cell stage, and the 8-cell stage, each half turned into a complete sea urchin. If Roux's theory was correct, it should have only ever formed half. Thus, he discovered that cells in the early embryonic period are self-organizing. He is thought to have made some of the first modern theories of epigenetics. Material outside of the nucleus influences what the genetic material in the nucleus does, which affects the outside of the nucleus, and you understand. This theory of reciprocal causation was totally new to science, but we see it everywhere now in negative feedback loops. Next are theories of cancer. The somatic mutation theory, SMT, says that cancer arises from mutations in certain genes called oncogenes. Mutations in these genes can occur randomly or in response to the environment. Cancer treatments are mostly based on this model, aiming at removing the mutated cells, so through surgical removal, radiation, and chemotherapy. Aneuploid theory says chromosomal abnormalities and rearrangements cause the progression of cancer. These abnormalities, called aneuploidy, interferes with the regulation of many genes, which leads to further interference with gene regulation. The picture here shows aneuploidy. The first and second chromosomes are the same in both pictures, so it's okay that they're cut off. But the rest of the picture is totally different because of the changes in the chromosomes, because of the aneuploidy. Both of these theories consider epigenetics as secondary, but we might have information to suggest that it's more important than previously thought. Characteristic methylation patterns occur in cancer cells. While cancer can occur from epigenetic mutations and genomic mutations, the epigenetic mutations are often overlooked and viewed as normal genomic mutations. Non-mutation based alterations in gene regulation are due to epigenetics. The last thing this book focuses on is called X inactivation. As we know, females inherit XX chromosomes and males inherit XY chromosomes. This seems unfair that a female would have more X chromosome to work with. Through a process called dosage compensation, females are actually only able to produce one X chromosome's worth of derived proteins. But this isn't a simple thing. We don't produce proteins from just one of these chromosomes. What happens is called X inactivation, where certain genes on each chromosome are, quote, turned off. This is a random process, so a female will express a variation of genes from the maternal X chromosome and the paternal X chromosome. It's like a stained glass window and another form of gene regulation. In closing, this book acts as an introduction to the complicated world of epigenetics. It teaches us how popular ideas like womb nutrients came to be and how many different ideas play into epigenetics. Epigenetics is any sort of gene regulation that does not occur through genetic alteration. It's an ever developing field, and new ties to other areas are always being found. Thank you. And for presentation purposes, here are my sources.